Sootybark disease, a new tree disease in British Columbia, is caused by the fungus Cryptostroma corticale. The disease was first found in 2022 in the Greater Victoria area and the Lower Mainland. It has been found on sycamore, bigleaf maples, and a few other maple species. Sootybark disease is associated with hot temperatures and drought in summer. The first symptoms include wilting leaves and branch dieback. Death can occur rapidly after the onset of symptoms, sometimes within a few months. There are several look-alike fungi and damaging agents that may be confused with Sootybark disease. To help accurately identify Sootybark disease in the field, researchers at the Canadian Forest Service developed a rapid and convenient diagnostic test. Here we see a dead tree that may have been killed by Sootybark disease. Underneath the bark, we see a layer of spores that we want to collect. If needed, we'll bat this bark to access it. Using tweezers, collect a few milligrams of spores into the collection tube. The solution will turn brown or black. Once all samples have been collected, head to the workstation to prepare the PCR. You will need an electrical outlet for this part of the protocol. To begin, rehydrate the PCR reagents. Add 18 microliters of molecular grade water into each tube. Prepare 1 tube per sample. Make sure to include a negative control consisting only of molecular grade water. Mix the reagents by flicking the bottom of the tubes and tap them on a flat surface. Let's move on to the samples. Shake each collection tube vigorously for about 30 seconds. The ceramic bead inside the tube will crush open the spores and release the DNA. Then, take a dipstick. Dip the wax-free end into the collection tube. Move the dipstick to the wash tube. Dip it inside to get rid of contaminants and cellular debris. Wipe off the excess liquid on the side of the tube. Carefully open the PCR tubes. Dip the dipstick inside. Make sure the end is fully submerged. Also ensure that droplets don't fall into neighboring tubes, as this can lead to contamination. Once all samples are added, place the strip tubes inside the mini PCR machine. Make sure to close it correctly. On your phone, go to the mini PCR app and click on the M symbol. Click on the mini PCR to connect it by Bluetooth. In the main menu, select the SBD protocol and click Start. The protocol takes about an hour and a half to complete. When the protocol is done, take the samples out. Check to see if you lost any volume to evaporation. Add 5 microliters of sample to each testing tube. Gently flick the tube to mix. In each tube, place a strip test with the arrow pointing upwards. When the solution reaches the buffer zone, take out the test and place it on a paper towel. Wait 10 minutes before reading the results. A positive result will have 2 bands, the upper control band and the lower positive band. A negative result will only have the control band. If no band shows up, the test is invalid. This test can detect concentration as low as 20,000 spores per milliliter, with an accuracy nearing 100%. From these results, we see that our sample tested positive, indicating that this dead tree is infected with Sootybark disease. This diagnosis can help inform urban foresters of the presence of the disease as well as the best practices for tree removal.